Hi, everyone. Welcome back to class. I don't know, whatever. Microbiology. We're going to get into chapter 11 this time. This is going to be talking about control of microbes. And what do I mean by that? I'm literally talking about controlling their growth. So what do we do on a regular basis to keep microbes from growing or affecting us in our day-to-day lives? Things like maybe cleaning your keyboard off. That would be an example, or cleaning bed off after a patient was there, or washing your hands, everything like that. Anytime you're trying to keep the bacteria away. Wow. So in this chapter, we're going to talk about the difference between sterile and other terms. We're going to talk about all the terms associated with this. We're going to talk about the physical methods to control the physical. So like the actual like scrubbing or something like that, the sudsing and stuff like that. The sterility and how we can achieve it. We're talking about chemical methods as well. So that's a different thing. So chemical method is like when you pour like a bleach or something on to something, that the chemical reaction is what is killing it. And we're talking about how those work as well. God, every time. Why? Okay. All right. So when we're talking about sterilizing, not sterilizing necessarily, but I think because I saw it on the first, but we're talking about microbial control. Thank you, Lauren. We can start with sterilization. That's an easy term. That's an easy term to be familiar with. If something is sterile, you guys know this, right? It's completely clean, right? There's no microbes. So this is destruction of all microbial life. Disinfection, however, destroys most of it. It reduces contamination on inanimate surfaces. So non-living thing. This is for non-living surfaces, like a tabletop or something. When we say that these disinfect most things, like these other things down here, is typically when a bacteria has an endospore. Remember how we talk about bacteria can form those to allow them to protect themselves from harsh environments. This protects them from this as well. So a lot of times it's the endospore forming bacteria that aren't getting cleaned by these things. Okay. So disinfection, inanimate surfaces, antisepsis. Let's call this degermination. I'm probably not going to ask you about degermination. Okay. But it's the same thing as disinfection, but it's on living surfaces. Then we have decontamination. Decontamination is just sanitization, making something sanitary. So that's going to be a mechanical removal of most of the microbes from an animate or inanimate surface. So if we're talking about like chemical removal of something, that would not necessarily be decontamination. We're talking about mechanical here. We're going to talk about like what that is in a moment. So here are some microbial control methods. We're going to talk about these and break them down. We have the physical agents, the chemical agents, and mechanical removal methods. Mechanical removal of something such as filtration would be decontamination. We just talked about that, right? In the previous slide. What I do want to point out with this chart, this is an excellent place to look whenever we're talking about these terms and what they mean. So when you're trying to determine, does this sterilize a thing or just disinfect, or is this antisepsis or what? This is a great chart to refer back to because it not only has, different lines that you can follow down to get to certain points, but also it has the actual terms over here. So please do refer back to this while you guys are studying. It's a useful chart. Primary targets of microbial control. We're going to want to get rid of microbes that are capable of causing infection or any spoilage. Like if we're trying to treat food to make it more user-friendly, like milk. Constantly present in the environment and on the human body, right? We want to get rid of all of that too, because we don't want it to grow. Like the stuff in my body that I'm touching your food, you don't want it to grow. And often when we're talking about any surface or whatever that we're treating, it's going to have a mixture of different kinds of microbes. So you want to be sure you're being broad. So how resistant can things be? We have the bacterial endospores down here. Those are the ones that are going to be the most resistant of the living ones. However, prions, those proteins that are misfolded, it's really difficult to get rid of those. You have to use extra high heat, higher than autoclaves, like 135 degrees Celsius would be what would be required in an autoclave. And we don't do that. That's 121 is usually what autoclaves do. And sodium hydroxide will do it, but pretty much nothing else. Mycobacteria are also pretty hardy. So that's like tuberculosis. That's an example. Staph does pretty well. We should know that by now, right? Anybody that's ever worked in any sort of hospital or had like anybody go to a hospital, you should know that staph is pretty hardy. MRSA is hard to get rid of. And so is C. diff. So C. diff would fall under the endospores because it's Clostridium. Remember, endospores, we're going to be Clostridium and Bacillus. So whatever. And then we can break it down even further down. But you see the viruses are going to be down here, non-enveloped viruses and the enveloped ones. Enveloped ones tend to be easier to get rid of. So sterilizing things. This is a process that's going to remove all viable organisms. We want to get rid of even viruses. I'm not going to get rid of prions, but that's not an organism. So we're not really worrying about that here. And they're super uncommon, so we're not worrying about that here. Material that has been subjected to this process is said to be sterile. That's been treated this way, and sterilized products are essential to our well-being nowadays. Our surgical instruments, syringes, and commercially packaged foods often have been sterilized completely. Disinfection, remember that's referring to a physical process or a chemical agent, which we call a disinfectant, that we use to destroy vegetative pathogens. So that's the ones that are not endospores. So endospores can survive this. We usually use that only on inanimate objects, inanimate surfaces. Antisepsis is that term that we're talking about on living tissue. So sepsis, that's a term to break it down. Sepsis is the growth of microbes the growth of microorganisms in blood and other tissues. Whereas asepsis is any practice that prevents its entry. So when we talk about aseptic technique, even in the lab, when we're in our lab, we're talking about sterile methods to exclude all microbes, as sterile as we can possibly manage. And anything that prevents contamination or cross-contamination is aseptic technique. It's promoting preservation of sterility. Anything that promotes the preservation of sterility. So antisepsis is application of chemical agents to body surfaces and surgical incisions to inhibit or destroy vegetative pathogens. Again, the endospores can survive. Right, so the agents versus the process is what we're going to get into. So what we call the agent and how it works. So the germicide and microbicides, these are things that kill all microorganisms, right? So that's what germicide and microbicide, so general use. Then we have things called bactericide, which guess what? That's going to kill bacteria. So these destroy bacteria, except for those in the endospore stage. Of course, we've already gone over this. You really can only achieve that was we were talking about with certain things. Fungicides kill fungi, virucides kill viruses, inactivate viruses because they're not technically alive. And then sporicides kill the endospores. So those are things that are capable of killing endospores. So you might have a chemical that doesn't work as well necessarily on vegetative, because remember the endospores are like a kind of different deal, right? They've got a different coating on them and all this stuff. So if something kills endospores, it might not necessarily work as well on vegetative. So you might want to mix the two. Bacteriostatic prevents the growth. So definitely know the difference between bactericidal and bacteriostatic. That's going to come up often. Bacteriostatic versus bactericidal, bactericide. So bactericide, kill. Think of like homicide. I don't want to say a bad word. So I have to put this on YouTube. I don't want it to get kicked off. Right, so bacteriostatic prevents the growth of bacteria. It does not kill the bacteria, it just prevents the growth of it in that environment. Fungistatic prevents fungal growth. Microbistatic prevents growth of microbes in or on the body. Something weird on my nose. Decontamination. we're talking about sanitization here. So it's the same word. It's an interchangeable term. Yeah. So it's cleaning technique that mechanically removes. I'm doing like this, like, you know what I'm talking about, like scrubbing, for example, actually physically, mechanically remove something, like filtering mechanically removes microbes. And then we have an example would be like anything that's a sanitizer. So soaps and detergents are used to sanitize. So they cause a sudsiness that causes the bacteria to not be able to stick on the surface of your hands. That's how it works. So when we are decontaminating things, we can refer to antisepsis or degermination, same thing, reducing microbes on the skin. We will often scrub or immerse it in chemicals or both. That'll emulsify the outer cutaneous layer to get most of the microbes off. So when you are in practice and you're learning about these things in a hospital, you might want to ask yourself what thing I should use to control microbes. So these are some questions you would want to ask yourself, right? Does the item in question require sterilization, or is just disinfection adequate? Do I need to get rid of endospores? Is the item to be reused, or is it permanently discarded after this? If it will be reused, can the item withstand heat, pressure, radiation, or chemicals so that we can go back and sterilize it if we need to? Is the control method suitable for a given application, like use on skin versus like you can't autoclave a person? And then will the agent penetrate the necessary to the necessary extent to remove whatever the situation is into the surface? Like, if you had a cloth, or if you had, you can't sanitize, rub me on my shoulder all you want, but you can't sanitize my stomach that way. That's what I'm talking about. Then the method, the cost, for producing it as well as using it and keeping it in stock and all of that. Yes, so we want to consider critical medical devices. So critical medical devices are the ones that we know are going to come into contact with sterile tissues. So like the deep tissues. We have also mucosal membranes that are not quite as sterile, right? That's not a sterile tissue, like in your sinuses and stuff like that. It's not a sterile tissue. We would use semi-critical. We will refer to these as semi-critical. And then non-critical medical devices are the things that are going to either we're not going to use them to touch a patient at all, or they're only expected to touch intact skin and not break the surface and introduce bacteria into. So what is microbial death? How do we measure this? This is hard to detect. We don't have any conspicuous vital signs. We would have to, how do we determine that something is actually been killed? Lethal agents do not alter the appearance. We can't necessarily look at the loss of movement, and because some of them don't move anyways. And then we have we'll have to develop these special qualifications to say whether or not something is dead, has actually been killed. So like if we're sterilizing something. The factors that affect the death rate of an organism, the number of microorganisms in a sample. If it's like highly overgrown, it's going to take a lot more to kill them all off. The nature of the microbes in the population, the type of the microbial growth that's going on. Is it biofilm, stuff like that? Is it bacterial, even stuff like that? Temperature and pH of the surrounding environment, concentration of the agent and how that could affect the environment or interact with other things in the environment, mode of action of the agent, and any inhibitors or anything like that. So how do antimicrobial agents work? These are the 4 ways they can work. This is a good thing to remember. So if we are talking about controlling microbial growth, these are for physical and chemical. These are the big 4. Okay, remember that there's 4. Cell wall, we're going to help prevent the cell wall from working properly, prevent the cell membrane from working properly, prevent the process of making DNA or RNA, and then getting rid of protein function. So the things that attack the cell wall, we're going to mostly be talking about things that block the cell wall synthesis so that new bacteria, as they're growing, cannot make new cell walls. The old ones are going to die. So these are things that will digest the cell wall as well. So we can either block it from being made, we can digest it down and break it down, or break down just like the surface of the cell wall. Anything like that is going to allow us to treat that bacteria or whatever more efficiently with other things. So the cell will be damaged more and lysed more easily. We can break it open more easily. So then things like detergents and alcohols will do this. They'll disrupt that cell wall. The chemical properties to do that. Okay, the cell membrane is the next thing. The cell membrane, remember on bacteria, we have a membrane, wall, membrane for gram-negative, and we have thick wall and membrane for the gram-positives. That's the typical ones. So keep that in mind. So the cell wall is important in bacteria. When we talk about the cell membrane, that's still, they both have cell membranes. They still have that inner cell membrane. This is what we're talking about disrupting with these. So these agents, we have cell membrane of the bacteria as well as the viral envelope counts in all of this. That'll be affected as well. We have disruption of cell membrane to cause loss of selective permeability. So we can't control what goes across anymore. Loss of vital molecules. We're losing things that we need to keep in the cell to the outside, and then allows the entry of damaging chemicals that we can introduce. So surfactants are things that are polar molecules with hydrophilic and hydrophobic regions. So remember when we were talking about that with phospholipid bilayers, they had a charge at the top, and then this part was hydrophobic, part that doesn't like water. So all the charged heads will come together, and then the tails come together, and then they would create like that on the same other side as well. That's what's depicted down here. That's a lipid bilayer. So these guys can mimic that. The surfactant molecules can mimic that and actually kind of intertwine into that and cause spaces to open up and cause holes in the actual membrane. So that's how surfactants will work. So they open up leaky spots, and that's dangerous, like we had just said previously, in those different ways. So how do we affect protein and nucleic acid synthesis? We all cells rely on a continuous supply of proteins. We need them to do everything. That's what your genes code for. So obviously you need a lot. So we want to inhibit ribosomes, because if you inhibit a ribosome, you inhibit protein synthesis, because ribosomes are necessary for protein synthesis. We can also inhibit nucleic acids. So if you want to impede DNA replication, so making new cells, you can't make more DNA. Or DNA transcription. DNA transcription is DNA to RNA. You can inhibit that. And you can also cause changes to the actual genetic code. When we talk about the native state of a protein, that's its normal functioning protein state. And then we have denaturing a protein. That's a term that you should know. Denaturing is talking about disrupting proteins in order to render them non-functional, changing their shape or binding with them or something like that. They can no longer do their job. So we can break the bonds that maintain their structures. That's often the most likely way that we're going to do it. So we can break it down into this. It doesn't work anymore if it's shaped like this. Nonsense. Agents that alter protein function, we have denaturation by moist heat. So here we're talking about like when you boil an egg. This is also what happens whenever we're talking about an autoclave. Other effects on proteins, we have metallic ions that can bind to the active site, so it can't interact with what it is supposed to interact with. And I've skipped right over organic acids and phenolics, but there you go, there they are. Heat is the most widely used method of microbial control. We have other methods as well. We have radiation, filtration, and ultrasonic waves, and cold. We can inhibit, control, or inhibit growth that way. We're talking about moist heat. We want hot water, boiling water, and steam. That's the most common ones that we're talking about. Temperature will range from 60 to 135 degrees Celsius. 135 Celsius is what I was talking about with getting rid of the prions. Moist heat, we can operate at lower temperature and shorter times than dry heat. And most microbicidal effects are going to be coagulation, denaturation of proteins, and halting microbial metabolism permanently. So literally killing. Dry heat, We're going to need a lot higher temperature. We're going to show you a little thing about that, but anywhere from 160 to several thousand degrees Celsius, usually using actual flames or heated coils. Because we don't have water being introduced there, it's hard to break up those protein bonds, so it doesn't work quite as well. So that's why we need the higher temperatures. The higher temperatures, and we have dry heat oxidizing the cells literally so that we can burn them to ash. So that's how that would work. That's literally like incinerator or something. So we have moist heat. We at 121 degrees, we can just sterilize it for 15 minutes. If we were to do that with dry heat, 121 degrees and no water introduced, we would have to do it for 600 minutes. I don't know if you have 10 hours to sit around and wait on something, but I don't want to do that. So you get the picture. Bacterial endospores, I'm doing this a lot. Bacterial endospores exhibit the greatest resistance to disinfection methods. Talking about we just mentioned this, the bacillus and the clostridium bacterium, right? So the endospores themselves, when they're vegetative, they're growing healthy, happy, doing what they should normally do. But and then spores are hunkered down and ready to resist everything. So how hard it is to destroy depends on the organism, and we often need temperatures above boiling in order to destroy them. So that's why we're talking about autoclaves with the 121 degrees Celsius. 100 degrees Celsius is boiling. So vegetative cells vary in their sensitivity to heat. The actual like non-endospore versions of them, it just depends on the bacteria that we're talking about. Okay, temperature and length of exposure should be considered for adequate sterilization. Higher temperature will allow for shorter exposure, obviously. So thermal death time is the shortest length of time required to kill all test microbes at that specific temperature, whereas thermal death point is the lowest temperature required to kill all the microbes within 10 minutes of exposure. So thermal death time, so it says it on here, the shortest length of time. And thermal death point is the temperature. So know the difference between those. So some common methods of moist heat control, we have steam under pressure like autoclaves. For non-pressurized steam, we have pasteurization, which is alternating of temperature, and then with repeated exposure to temperature, and then boiling water. So when we talk about steam under pressure, we're getting into the autoclave territory. If you don't know what an autoclave is, it is a large thing that can hold a lot of pressure, and it builds up pressure and puts steam in, and that pressure and steam chamber can get it up to 121 degrees Celsius, and we put it there for 15 minutes and 15 PSI, and you can kill pretty much anything within that amount of time. That's the idea with an autoclave. Okay, it's like a home pressure cooker, like if you have an Instant Pot. Okay, so the steam, however, has to contact the item's surface in order to work. In this one, this guy was wrapped up in the foil. When they put it through the autoclave and they took it out, the steam hadn't touched it, so it was not sterilized, whereas this one that was left out was. It's important that you keep it exposed to the steam, otherwise it's not getting treated. At what temperature does an autoclave achieve? At 15 PSI. PSI is pounds per square inch. It's a pressure measurement. So 15 PSI is going to achieve 121. We already talked about that. So intermittent sterilization is also known as tyndallization. So that's just tyndallization. If you see that word, just know intermittent sterilization. So we have free flowing steam for 30 to 60 minutes, incubate it, let it sit for 23 to 24 hours, and then treat it again. And you'll do that 3 days in a row because one exposure isn't enough to kill. So we use this for some canned foods and certain media that's special, and we can't just treat it with autoclave. So pasteurization is a sort of similar thing, but faster. So we have heat applied to liquids to kill potential agents of infection and spoilage, right? You guys know your milk has been pasteurized. If you buy lactaid milk or the lactose-free milk, those milks have been ultra pasteurized, so they last way longer. I don't know why people drink regular milk. So heat is applied to liquids to kill them. We have special heat exchangers that will basically wash the liquid over. The flash method, in this case, we're going to expose it to 71.6 degrees Celsius for 15 seconds, and then off, and then on, and then off, and then on, and cooling and heating and cooling and heating. And then the batch method exposes it to 63 degrees Celsius or up to 66 for 30 minutes. So yeah, so then it'll expose it and then, you know, cool it down. So this does not kill endospores. So endospores are going to make it through, or anything that can resist heat won't be killed by this either. So pasteurized milk is not sterile. That may be a question on your test. Pasteurized milk is not sterile. It has bacteria in it. That's why it goes bad so fast, guys. Boiling water, we will use this for disinfection, not for sterilization. We'll expose materials to boiling water for 30 minutes at least in order to do that. When we use dry heat, moving on to the dry heat. Incineration, in this case, we're talking about igniting and reducing microbes to ash or gas. We do this in the Bunsen burner. You guys do this with your loops, right? We have a dry ovens. You guys are familiar with ovens. I feel like I don't need to explain how that works, but you have a coil that heats and it exposes things to the heat for a while. So we do this in science with glassware and medical instruments sometimes, if they don't need to be sterile. So which of these is likely to result in sterilization? Of all of these, we can, least likely, sorry. I was like, so this is true. This is true. No. Yes, so this could work depending on the organism, depending on how long we're putting it in for. Autoclaving will work. That's for sterilization. So the answer would be boiling. We already said boiling doesn't sterilize. Well, it didn't give you the answer, but it is that. So cold treatment retards the activity. So I'm talking about slows the activity. So in order to slow the activity of that organism, you will have to get it to a certain temperature that actually works for that organism, but it's not going to be dead. You still have to get the thing out of the freezer and then cook it in order to make it safe. So desiccation is talking about, sorry, I was reading the slide and I wasn't saying anything. Desiccation is dehydration, essentially, right? So just drying a thing out in order to kill things, but it doesn't always work. Not everything can be killed that way, but you can preserve foods that way. It is a little useful that way. Preserve, but not kill. Lyophilization is just drying out at freezing temperatures. So getting rid of all the moisture at a freezing temperature. You often have vials that are filled with powder, they have the little rubber stopper in, and you have to put the like liquid into it to mix it up. So that's lyophilized. Radiation, we can use that to kill things. Typically, when we talk about radiation, we're talking about anything from UV over through gamma rays in order to kill something. Ionizing versus non-ionizing. Irradiation refers to bombarding microbes with radiation. So particle waves, that's what radiation is. Ionizing radiation, that is a type of radiation where we are ejecting orbital electrons from an atom, like the actual electrons are flying out of it. And then that will cause ions to form from that atom. So that atom is turning into ion version. Remember what happened whenever we would have like sodium and chloride and they'd separate and we have ionization, ionic bonds between that, right? And then we have ionization, but this is happening because the frickin' atom is just kicking out its own electron. So this is going to cause the most damage to proteins because this is going to literally physically like have electrons fly out at the proteins. That's what's going on. Non-ionizing radiation is going to excite atoms and just literally like causes abnormal bonds to form where there shouldn't be bonds. And that's pretty common in DNA for that to happen with exposure to UV radiation. So this is examples of ionizing versus non-ionizing radiation, not examples, but like how dangerous they are comparatively. So we have this radiation source of whatever is exuding the radiation. With ionizing radiation, we're talking about it being super dangerous. It can get through barriers, takes very special barriers in order to protect from the ionizing radiation. And so that'll still get through that barrier to the cell and the DNA. And then that will cause breakage due to loss of the electrons being expelled out from that radiation contact, turning it into ions. Then non-ionizing radiation, like UV damage, then we're going to have abnormal bonds forming. You can't really see it as well. Like you see it on that like little red like square that's formed. That's a bond next, like the red next to the red bound to it instead of across where it's supposed to be. So that's what's happening there. Yeah, and UV doesn't penetrate barriers. Moving on to the use of radiation in microbial control. You can use it for cold sterilization. We don't have to heat something up in order to sterilize it. We can use ionizing radiation, just treat the whole thing with ionizing radiation. It'll be an effective alternative for sterilizing materials. And we use this for materials that are sensitive to heat or chemicals because it doesn't cause any damage that way. Then we have ultraviolet radiation. We can use these in the medical area. Typically, it's most lethal from 240 to 280, and often see it at about 260, 260. Whenever you're putting lamps up. So here we have a germicidal lamp example. You turn it on into one of these hoods here and leave it on. We'll leave it on for like 30 minutes or whatever, and it will kill everything that's on, contaminating that surface. Typically clean it with like another cleaning compound as well, just to be double sure. So non-ionizing radiation, moving on with more of this talk about UV rays. This is the sort of what I was talking to you guys about. We're talking about pyrimidine dimers, specifically thymine. So that's the T's. So the T's, thymine dimer, di 2. So we have two T's stuck together where they should be binding across to the A's. And they can't do it well. And so this causes problems in DNA function as well as replication. Can't make copies anymore. We're going to filtrate. Now we're talking about decontamination by filtration. So a mechanical method to remove microbes from air, like HEPA filters, or liquids. Fluid is strained through a filter with openings large enough for liquid to pass through. Yeah, so I don't know. So yeah, HEPA filters, that's what they do. That's what they're for. That's why they're special. That's why they go on and on about them. Yeah. So you can even filter out microbes or viruses with that. So we can use filtration for liquids that can't withstand heat. That's what kind of why it's a similar idea with the coffee. We can pretend that the coffee grounds are I said coffee because I saw this vacuum cleaner and I was like, man, that makes me think of coffee. I guess I must need coffee. But anyways, talking about coffee, it's a similar idea. Thinking of maybe your bacteria being the grounds, and then catching it in the filter. That same way. But the liquid is still getting through. So, and then in this case, they're showing the vacuum cleaner because of the HEPA filter that's been on it. So most vacuum cleaners nowadays have HEPA filters to keep us from shooting bacteria and everything out into our environment. Yeah. So filtration, it's another way to clean out the air as well as liquids. So which of these would be least likely to be removed by passage through a HEPA filter? The smaller, the less likely they'll pass through. So protists, protozoans are eukaryotes. This is a eukaryote. So eukaryotes are typically larger even than bacteria. Bacteria are typically larger than viruses. So we're mostly going to be talking about viruses here. They're the tiniest. Moving on to using salts and sugars, that's creating that hypertonic environment, causing plasmolysis, right? So it's all the sugar or the salt on the outside of whatever it is, drawing all that moisture out of whatever bacteria could be on there, drying out the bacteria and killing it that way. So that's how that works. That's osmotic pressure. So yeah, sugar and high jams and jellies work the same way as the salt does. And so chemicals, moving on to the chemicals. Jeez, man, there's still like a lot left. I didn't realize how long this was going to be. Okay. Moving on to the chemicals. We have solid, liquid, gaseous. We can treat things with gas. I feel like some people forget about that, but yeah, a lot of different ways we can treat things, different forms using chemicals. So we have the aqueous solutions that are based in water, and tinctures, that's a term you should be familiar with, that is mixed in alcohol or alcohol water mixture. So here's some examples of some typically used agents. When we think of chlorine-based ones like bleach, or we think of alcohols and whatever and how they work, if you're interested in looking over this for how toxic it is to people versus special comments over here, how active it is and successful it is at killing things. So, and what microbes it targets. So when you're selecting a microbicidal chemical, you want things to have rapid action. You want to have to use very little of it with rapid action, soluble in water or alcohol. It's long-term stability, broad spectrum, doesn't have any toxicity to humans or animals, penetrates inanimate surfaces so that it can have persistent action, resists inactivation by organic matter like pus or urine or whatever, blood and whatever. It's non-corrosive or non-staining. And sanitizing and deodorizing properties to keep everything pleasant for like patients who might be coming in afterwards, and affordability and readily available. You don't want it to be too expensive, and you don't want to have to be like importing it from, I don't know, Russia or something. So things that are high level germicides, these are the things that kill endospores. Chemicals that kill endospores. It's not physical stuff anymore, not mechanical. They will sterilize if they're used properly, and they are critical items that are not heat. This is for things that are critical that are not heat sterilizable. So you would do this to treat things like catheters or heart-lung equipment, to treat it to make it as sterile as possible, because you can't heat sterilize these things. The intermediate level ones, these ones kill fungal but not bacterial spores. So bacillus and clostridium will still make through, but the fungal spores at least will be taken care of. They also don't kill resistant pathogens like tubercle bacillus, and which is talking about tuberculosis, and viruses, certain viruses. So these are used to disinfect semi-critical, my nose is really tickling, I'm sorry. Semi-critical items, not the ones that have to be sterilized. So respiratory equipment and thermometers, things that you would be reusing, but don't have to be sterile. So these are the low-level germicides. They will eliminate vegetative bacteria or vegetative fungal cells. All the spores will get through though. Some viruses can be killed. These are used to clean non-critical materials like electrodes, straps, maybe the rubber mattress cover thing, and all of that. What affects the chemicals has to do with the nature of the microorganism being treated, the nature of the material being treated, like if it is one of those covers of the mattresses, or if we're talking about, the skin or something like that, the degree of contamination, how contaminated is it, the time that it was exposed to the chemical, and then strength and chemical action of the germicide itself, how it works. So germicides can attack all the same four, the big four, right? Proteins, nucleic acids, cell wall, cell membrane. It was this, and then this on the other one, but forgive me here. So we are going to break it down into the different chemicals now. Halogens. Halogens, it's literally the halogens on the periodic table of elements is what we're talking about. Fluorine, bromine, chlorine, iodine. These are microbicidal and sporicidal, and they have active ingredients in about 1/3 of all antimicrobial chemicals because they are effective. So we have gaseous chlorine, Cl2. It's pretty toxic to us. We can't be exposed to it. We have hypochlorites. That's like bleach. We have chloramines. It's usually a combination of, we have the amine group as well, but that can also interact with the alkaline pH. So it has a lower hydrogen concentration, a higher pH. So, yeah. It's used as an alternative basically for chlorine. Then we have the iodines. These are like iodophores, anything that has iodine incorporated with it. So these are typically not adversely infected by organic matter. So that's nice. And we can use these on skin if you're not allergic. These are some lists of them. Aqueous, iodine tincture, that's with alcohol, tablets, you can put it into your water to make it drinkable, and iodophores are a mixture of iodine and alcohols, and complex with other things. And then that could be used for medical antisepsis. So we would be talking about like skin. So phenolics, I like how they smell. These are used as, this is like chloroseptic. That's a chloroseptic that you spray in your throat. That stuff is a phenolic type of thing. So I don't know, I like the way it smells. Or like old school Carmex. But yeah, anyways, it was the first one that was used as an antimicrobial chemical, and it is toxic and it is irritating. So it's not great to use it anywhere near skins, but it can be used near skin. Phenolic, so we have phenol itself literally, and then phenolics that are based off of phenol in their structure. These destroy vegetative bacteria, fungi, and some viruses. They can act in the presence of organic matter, but they're too toxic to use as antiseptic, so we don't use these on skin. Then we have cresols, bisphenols, triclosan might be one that you guys were familiar with. This used to be like that antimicrobial that was in like hand sanitizers and stuff like that they discontinued because it was contaminating the environment. Then we have chlorhexidine that they use for hand scrubbing, surgical prep, and other medical uses. If you guys haven't heard of chlorhexidine and you work in a hospital, I don't know what you're doing, because they use quite a lot of chlorhexidine in hospitals. It's very popular, but it is a phenolic. So alcohols as antimicrobial agents, isopropyl alcohol and ethanol are the ones that we use. They have the best, they work the best at 70%. You need a little bit of water in there for them to work. It's sort of like, not quite like the steam, but just think of it the same way. So it needs a little bit of water to work. Vegetative, but not endospores, it'll kill. Applying, using the alcohols. We can use alcohols, ethyl alcohol for skin degerming, that's a weird word, degerming and disinfection of some types of medical equipment. And then it has a evaporation rate that's pretty high. So does isopropyl alcohol, but it's more microbicidal, works a little bit better to kill things, less expensive as well. But yeah, you can get pretty bad adverse effects from the isopropyl alcohol. I had that happen one time by accident, cleaning the floor with it, and it didn't work out so well for me. Hydrogen peroxide. You guys heard of this? H2O2 is its chemical form. You should probably be aware of that, because I'm going to be using that a lot in the lab when we talk a lot about hydrogen peroxide in the future. But H2O2, okay, hydrogen peroxide. There's a hydrogen per oxygen. That's what it means. So it has germicidal effects because it is creating a toxic reactive oxygen that kills bacteria. It's bactericidal, virucidal, fungicidal, and sporicidal at high concentrations. It's pretty useful. So what do we use it for? We can use it as an antiseptic and skin care, like cleaning skin, wounds, bed sores, mouthwash. Even says it on the bottle. If you buy it at like CVS, you can use it as a mouthwash. We use it to disinfect things such as surgical implants, sterilizing delicate reusable instruments at the high concentrations. We can vaporize it into a gas for small industrial sized items that we need to get into. And then ozone is similar. It's a toxic oxygen as well, has similar effects as hydrogen peroxide. So, you know, same kind of thing. Surfactants, we already talked about these a little bit as far as their mechanical effects go, but as far as their chemical effects go, surfactants have limited microbicidal power. They usually work due to their amphipathic nature of the molecule. So remember that charged head and the hydrophobic tail. They have varied effects, depends on what you're talking about, and activity is reduced by the presence of organic matter. So this is just a picture of like what you would see. Here we have the hydrocarbon chain. So this is the hydrophobic tail. And these are examples of like the charged head. So this one's positively charged head. This one associates with chlorine, but yeah. Benzalkonium chloride, for example. So this is going to be things we're talking about now, moving into the quaternary ammonium compounds called quats. These are disinfectants that can be mixed with cleaning agents to sanitize. They are preservatives for ophthalmic solutions and cosmetics like your contacts. And then we have, what is touching my leg under here? Soaps. They be used in soaps. So this, if we talk about quads and we're talking about, what's what I'm trying to say is the detergent. So we're talking about this. This is quaternary ammonia compounds. And why am I calling it that? Because it's N and it's got all these 4 things that come off of it. N, 1, 2, 3, 4. That's 4. That's quaternary. And it's ammonia because it's the N bound to 4 things. So soaps, next thing, detergent. So they aren't very microbicidal. Pseudomonas is an example of a bacteria that grows on soap dishes. And yeah, mostly used for mechanical removal and not really anything else. And we usually typically combine it with chlorhexidine or iodine in order to make it useful. So what kind of property do surfactants have? They are amphipathic. Remember, they have the charged head and the hydrophobic tail. Heavy metal compounds, moving on, we have mercury, silver, gold, copper, arsenic. Is that what this is? And then zinc, I think it is. Anyways, oligodynamic action, meaning they have antimicrobial effects in exceedingly small amounts. You don't need very much of them in order to work. We know this a little bit about silver. It's become pretty popular to have silver incorporated into things so we can make it antimicrobial. Drawbacks can be toxic. They can cause allergic reactions. If you have a lot of bodily fluids involved, it can neutralize their action, and microbes can develop resistance to them over time. So essentially they interact with the proteins to make them so that they can't work properly. So just put silver onto this plate and then put bacteria to grow. And then you can see from silver, and this is gold over here, it's inhibiting bacterial growth around it. See, mercury, they used to use tinctures of mercury for antiseptics. Not really that great for putting it on your body, I don't feel like, but you can still use it. They use weak organic versions of this, very weak, not very much mercury in it because you want to, but they do use it as a preservative. Silver compounds like silver nitrate, sulfadiazine. If you've ever had a burn, you've probably been treated with that. Using pure silver that you can put into the catheters, like embed it within the catheter itself. Colloidal silver, and then you can mix it into a solution. And then silver ions we can incorporate into hard surfaces and textiles, make it antimicrobial. Like figs, I think, are scrubs that have something like that. I don't know. Okay, aldehydes. Moving on to aldehydes, we know these are dangerous for killing anything. That's the whole reason that we use them in preserving bodies, because of their antimicrobial properties, right? So glutaraldehyde, formaldehyde, and phthaldehyde. Yep, OPA. Anyways, broad spectrum. Glutaraldehyde can sterilize things, and it works in the presence of organic matter. Yeah, so you can use it to sterilize things that you can't put into the heat. Formaldehyde, we can make it into an aqueous solution with water, and that becomes formalin. It can be an intermediate to high level disinfectant, can become relatively close to sterilizing, and then it's extremely toxic for people, so you don't want to be around it too much. And OPA is a little less irritating, but it doesn't destroy endospores. The gaseous sterilants, things that we might want to put things into a room, large things to sterilize them, we would use the ethylene oxide. So that's what we call a chemiclave. It's like an autoclave, but using chemicals instead of the steam. So ethylene oxide is explosive. causes damage to lungs, eyes, and mucous membranes. So you don't want to use it too much around people. And chlorine dioxide is also something that is used. They used it to disinfect all of Congress after the anthrax attacks in order to make it safe for people to be in there. So ethylene oxide, when we're talking about this just now, I don't know why it's on here twice, but whatever. It sterilizes and disinfects plastic and delicate hospital instruments. So things that can't be exposed to heat, like a lot of this. And then propylene oxide is less toxic than the ethylene oxide. And so we'll use it often for foods as a gas to treat them. If not, anyways, dyes. So we can use some dyes are used to sterilize, not sterilize, but as antimicrobial agents. They have just that effect on some things, but they're not great because they stain the skin and they're not very, they're usually very narrowly effective. Next, we have the acids and the alkalis. So we can have an aqueous ammonium oxide. So we talked a little bit about ammonium earlier, the quaternary ammonium compounds, the quats. So we can incorporate the, less, I don't know, complicated versions of them into detergents, cleansers, and deodorizers. Then we have organic acids. So the ammonium ones, these are the alkaline ones. And then these are low pH, the acids. So these are acids. We use them for food preservation. Acetic acid, that's, remember, acetic acid is vinegar. So things that we preserve in vinegar. I'm sure you guys have bought things like that. And whatever other acids you can preserve things in. So triclosan for a while was being excreted in the urine of 75% of Americans. Yes. And we were just getting it all over ourselves and using it for everything. And we were just, it was all over the place. It was detectable in levels in certain groundwater sources, and it would create dioxin-like compounds when exposed to sunlight. That was kind of a bad thing that would be toxic to us. So, and to the environment. Yeah, and it promoted resistance to triclosan and other antibiotics that were related to it. So we did stop using it. We don't use it anymore for a very big reason. So anyways, these are some products that people use kind of regularly. So if we look at Clorox disinfecting wipes, it uses dimethyl benzyl ammonium chloride, and that is a quat, which we talked about earlier. I'm on that ammonium and then other crap stuck to it. Yeah, dimethyl benzyl. So you can kind of look down the list and see, oh yeah, I know of this. I know Renew contact lens. I have that. Well, it has this propylaminopropyl biguanide, and that is a chlorhexidine derivative. Interesting. So whatever, if you want to look through this, there is that. And now that you know a little bit about the things that you're putting on your body or putting in your household, maybe you have a little bit better understanding of what you're going to be getting into whenever you start cleaning things around hospitals and whatever, if you haven't already been doing that. So what we went over in this chapter, this was chapter 11. This is just an introduction into unit 3. And it's going to get, this is the easy chapter. So we have terminology that we introduced as far as referring to how we clean things, death rate of things, that thermal death time versus the death point, physical control versus chemical control with things. And we went through each of those. So remember those big 4 being the big 4 of how we treat things: attacking the cell wall, membrane, the DNA functions, and the proteins themselves. So those are your big 4 of what these things are going to be attacking. So do remember that. And I will see you guys for chapter 12.